Have you ever wondered what the microbiome of your reef tank is? What even is a microbiome? You know, the bacteria, the microorganisms that you can't see in your reef tank that's doing all the work. Well, today I have Dr. Eli from Aquabiomics to share a little bit more info and insight into that world and what Aquabiomics actually is and what the microbiome is. So, welcome to the stream, Dr. Eli. How are you?
Pretty good. How about you?
Doing well, doing well.
Thanks for the chance to talk about aquarium microbes with you.
Yeah, absolutely. Thank you for being here on the channel. So, diving right into it, what is Aquabiomics for anyone that's new to
Yeah. So we're a brand new startup company offering the first DNA testing service for the aquarium. And this testing service encompasses both the microbiome, that is the microbes that live in your tank, that's the bacteria and archaea, and it also encompasses other organisms that live in your tank, things like parasites and nuisance algae. So we have the microbiome test for the bacteria and the tank DNA test for everything else.
Gotcha. And so with these tests, you're able to, I guess, see into that world of like the nitrifying bacteria and beyond. So us as hobbyists, we know about the nitrogen cycle. Ammonia converts to nitrate, but you actually can see bacteria that's responsible for that.
That's right. Yeah. So that's a great example of the good kind of bacteria that we're hoping to find in your tank. And that's one of the reasons to use one of these tests, is to find out, do I have the microbes in my tank that I want to have in my tank in order to have a successful reef ecosystem? And of course, the flip side of that is another reason to take these tests is to figure out, do I have any of the bad bacteria, the pathogens that may be causing diseases for fish or corals in my aquarium? So the test really shows you just all of the microbes, all of the bacteria and archaea that are in your sample.
Very cool. And so if you're new to reefing, so I guess when would be a good time to send in a test? Like right when you first set up a tank to get like a baseline, or let it sit a few months or a year, or wait till you see a problem, or
Right. Yeah, it's a great question. You know, as the guy selling the tests, I guess part of me wants to say, yeah, take a test every week, right? But let's be honest, it costs some money and it takes some time. So let's think about how to use the tool intelligently for reefing. You know, I think about it kind of like a blood test in human medicine. It's not something you need to do certainly weekly. It's not something you need to do every day, but it is good to get one routinely. You kind of described one reason for that is establishing a baseline. You know, if your tank is doing well and you want to know, what does the community look like when my tank's doing well, go ahead and get a baseline. And then later, if something's gone wrong in your tank, you can take another test and compare them and say, well, has my microbiome shifted into some community that doesn't look like a healthy community? So yeah, it's good to take one as a baseline. It's good to take one when things go wrong. I'm not sure I'd recommend taking one the first day you start up your tank. I'm sure we'll get into some of this, but the microbiome develops over time in a new tank. And unless you're interested in doing an experiment, I can tell you right off the bat that on day one, especially if you've started with dry rock and, you know, sterile dry sand, you're going to have, you know, a very immature microbiome that's not ready.
Mm-hmm.
So I wouldn't, again, unless you're doing an experiment, I wouldn't spend your test on that day one sample. Instead, I would do something like wait a month or so until you think the tank is ready, and then take the test to evaluate that question. You know, is my reef tank microbiome mature? That's actually something we can evaluate based on what we see in your test.
Yeah, because there's I know with bulk reef supply, they're testing multiple tanks and setting it up with dry rock, live rock, live sand, all the different variables that go into setting up a tank, and they're trying to see like what's the best way to do it.
Yeah. So that in their case, you know, we're taking samples all throughout the experiment, right? Because we want to see that change over time, and it's turning into a really nice data set on the development of the microbial community over time. It's great to have these data combined with all the videos that they're taking, so you can see that, you know, here's what it looked like at the beginning, and here's what it looked like when it was mature.
Yeah, absolutely. And so what's like the goal of Aquabiomics? Because you're collecting all this data. How many tanks or tests have you taken so far, or like what's the end goal for it?
Yeah. Well, so I mean, to directly answer your question, the goal of Aquabiomics is not primarily a research goal. It really is primarily to provide this service to the hobby.
Very cool.
You know, I started off as a coral biologist. So I spent 20 years in academia studying a variety of different things, but always using DNA sequencing technology. I kind of fell in love with those technologies early in grad school and carried them all through. And in my time studying corals, like many coral biologists, I kind of struggled with getting corals to grow well in the lab. I think your audience will appreciate that. You know, it's not an easy task to get corals to grow in an aquarium. It's not just a case of pop them in the aquarium and everything works. You know, and we had a lab at our disposal. We were measuring everything you're supposed to measure, right? Again, I think many of your listeners will sympathize with that experience. We in the hobby spend a lot of time measuring things like alkalinity and salinity and temperature. And, you know, we try to get everything right. And yet we have a tank where corals aren't doing well, right? That really was the motivation that drove me to develop this, because I knew that there were other things we could be measuring. You know, I was using these DNA sequencing technologies and my colleagues were using them. And I knew that there was this whole world of you know, the marine microbiome that wasn't being measured in aquariums. And since the service wasn't out there for the hobby, it wasn't out there for me either as a researcher, right? So I said, okay, somebody has to develop this service. So that is our primary goal. With that said, a part of that service has to be research, because you know, one of the great challenges to this kind of work is that most of the bacteria that we identify in your sample, we won't be able to point to a specific paper that says, here's exactly what that bacteria does. Most of the bacteria in the ocean, most of the bacteria in your tank are not well described. You know, we don't know exactly what all of them are doing. And so there's this big knowledge gap, right? We need to fill that gap in our knowledge. And the work we're doing at Aquabiomics is helping to fill that gap. Really in collaboration with the hobbyist community, because you guys are the ones out there doing the experiments in the tanks in your living room. And we're just the ones coming in to provide the service, the DNA testing service to help analyze your experiments. So we do have that additional goal of learning more about the microbial community in your tank. And we can get into some of those details today.
Yeah, absolutely. And that's very cool, because in the hobby, like every tank is different. However, there's a lot of similarities with on the bacteria end, and we'll take a look at that when we look at the sample that I sent to you and take a look at the results. So very cool. So I just want to show this clip right here. This is a clip of the tank with some dinoflagellates in the tank that I was battling. And I believe I sent the test off to you shortly after gaining control of the tank again, I guess, and kind of fighting them back a little bit.
It's a beautiful shot of dinoflagellates. Yeah. So I'm pulling up your test results. I can't recall which type of dinoflagellates we found DNA from. Yeah, it looks like it was Amphidinium, which was consistent with what you were suggesting based on what it looked like under the scope.
Yeah. And I used just a small little handheld microscope, and this is the shot that I got of it. And based on some images I found online comparing the two, that's what I came up. Looks like Amphidinium to me. So
cool. Yeah, I'm terrible at interpreting microscopy. I'm much better at interpreting DNA sequences. But at a DNA level, yeah, it certainly look like Amphidinium. Along with diatoms, which is something we often see in in
Hmm.
nuisance algae situations, it's often not, a pure culture of a single thing causing the problem in your tank. It's often a mixture of various
Combination.
things.
Yeah.
Yeah.
And in the case of my aquarium, it's about a year and 8 months old now.
Okay.
However, it was set up with water from a reef tank that was established for 5 years.
Okay. Great.
So I basically just transferred all the water, all the rock, and everything over to this tank.
Yeah.
Um, so I noticed with that tank, like, of course, day one, you put everything in. Once the cloudiness goes away from the sand, looks nice and pristine, and
Mm,
then of course those dreaded ugly phases comes back, and cyanobacteria,
sure.
dinoflagellates, hair algae, all that kind of stuff.
Yeah.
Um, is there a way, knowing what a healthy microbiome looks like, is there a way to kind of skip those phases, or?
Yeah. It's an important question. So, yeah, I think we've all encountered that, right? Probably in
Mhm.
every tank that any of us set up. Um, my my position is, after having to set up tanks a lot of different ways and measured their microbiomes, my position is you can probably never avoid the uglies altogether. We'll talk about why in a second, but I do think you can minimize them greatly by inoculating the aquarium with a nice diverse population of the appropriate bacteria. And it sounds like you did exactly that. Taking rocks and water from an established healthy tank is a great way to seed the microbiome in a new tank. Um, the reason I say I'm not sure we can ever avoid it completely is, think about, in a new tank, there are some surfaces that are truly clean. Whether it's the glass or the plastic pipes, or pieces of sand that previously were not exposed, there's surfaces there that are truly clean. They don't have a bacterial biofilm growing on their surface. And there's really kind of a succession process that happens on any surface when you put it into water, especially seawater. Um, Different organisms start growing on it, and you go through different phases, right? You go through the early phase of some organisms, some kinds of bacteria, and then different types of bacteria take over. um, it's mostly invisible in the aquarium until you get to the ugly stages, and then you can see the visible ugly nuisance algae. um, so I don't think we're ever going to avoid it altogether, because we're always going to have those naked, clean surfaces. But by putting the right microbes into the tank in the first place, it does seem we can greatly reduce it, almost to the point where, it almost doesn't happen. It's not a major problem in some tanks.
Yeah, very cool. And yeah, and so as far as the testing, I found it very easy, very straightforward.
Cool.
Um, Here is a video clip of me going through one of the tests. But um, yeah, so you receive, if you're doing both of the tests, you get a box for either the microbiome or the DNA. And so again, what's the differences between the two
Yeah.
kits?
Yeah. So the sampling procedure is pretty similar. And this is awesome. You have a great video of it for us to look at while we talk about it. So for the microbiome sample, you're really taking two samples. You're going to take a water sample, and that's collected using the syringe that you're seeing there. And you're also going to take a swab sample to collect the bacterial biofilm. In the tank DNA test, there's just a water sample. There's no swab. We're just collecting DNA from the water. Now, there's another kind of hidden difference that, as the user, you don't really see, but it's happening in the filter. That is, when you take your sample for the microbiome, your goal is really to capture bacteria and archaea, cells that are floating around in the water. And so you're capturing those cells and filtering them, pushing them down onto a filter. So the cells themselves, here we are drawing the cells out of the aquarium, and then you're going to push them onto a filter and capture the cells. Uh, and it's the DNA in those bacterial cells that we'll be analyzing for your microbiome test. um, in contrast, the tank DNA, that test, it looks just the same when you're sampling it, but our goal there, it's actually a different filter. our goal there is not to capture cells, but rather to capture the DNA that's floating around in the water. So we use a DNA binding filter. It has bigger holes in it, so you can push more water through it. But overall, the process is very similar for both of them. You collect your water, you push it slowly through the filter. And thank you for showing this. This is great. This is a challenge that I think some users have. They try to push it through too quickly. And if you apply too much pressure, you'll blow out the seal and lose your sample. So take your time with it. This looks great. Could maybe even go a bit faster, but it never hurts to go too slowly. The goal is to collect it without blowing out the
because when you're pushing down, there's definitely a lot of pressure getting it through the tiny opening at the bottom, for sure. And so so that DNA that you're collecting, that's known as eDNA, right? Or
that's
environmental DNA.
Yeah, exactly right. So in the tank DNA test, we're collecting eDNA, environmental DNA. Most of it is actually dissolved DNA just floating around in the water. But there's also some chunks. There's some cells floating around. There's some pieces of tissue floating around. And the microbiome test, in contrast, is really analyzing bacterial cells themselves.
Very cool. So I imagine that you find all kinds of things in there. You probably get this question a lot, like what's the strangest thing that you've found in the DNA sample?
Yeah, boy. I mean, we find all kinds of stuff. In the it's best to talk about that question in the tank DNA test, because the organisms we find will have names we recognize, right? Mm-hmm. So we find in the tank DNA test all the animals that you would expect to find in a reef tank. We find fish and corals and snails and worms. Of course, none of those animals made it onto the filter, but their DNA makes it onto the filter. Mm-hmm. But we also do find some strange stuff in there. One thing we find in almost every sample is a human skin fungus with an unpronounceable name. It doesn't really matter because it's not a pathogen. It's just a part of the community that lives on all human skin. But if you look it up, it's like, yep, this is clearly a human skin fungus.
Oh, wow.
I sometimes think that if there's a human skin fungus researcher out there, they should get a hold of our database, because we have samples of skin fungus DNA sequences from all over the country at this point.
Interesting.
So that's an example of something we find commonly that definitely didn't grow in the tank, right? It was a contaminant that got into the tank. And another one like that, we find terrestrial plants. One guy's sample recently had a whole bunch of cotton. I imagine it must have been a cotton field near his house. Pollen was probably blowing in.
Interesting.
So we can identify not just, it's a plant, but here's the species of plant that we found in your tank. What other things? Occasionally we find terrestrial insects, a spider or a bed bug. We found bed bugs in a couple of samples. It's never a lot of the DNA, right? It's always a tiny, tiny fraction of the DNA. But we certainly do find, odd contaminants. And that's without even getting into the kind of unexpected marine organisms that we find from, unusual invertebrates like bryozoans. These show up sometimes. Ascidians, another invertebrate group, shows up sometimes. So those are kind of unusual ones that show up. And of course, there's unexpected parasites show up sometimes. They weren't on the list that we were looking for in the hobby, but we find it in the DNA anyway. And that's, in my mind, one of the real great advantages of using this kind of technology for this application. If we were doing qPCR, which is the way everybody got COVID tested, they did a test on you to ask, do you have COVID? It didn't tell you if you had some other virus. It just gave you yes or no for COVID. But if you use DNA sequencing, you get to find out everything that's in there, whether you were expecting to find it or not.
That just fascinates me how much you can see with like a small sample of water. It's really amazing.
They're using it in research. I was inspired to branch out from the microbiome to tank DNA based on what they're doing in research. So they go out and sample from the ocean on cruise ships, and sample some water, and they learn about the fish that are living in that part of the ocean. Like the ocean's huge compared to
Yeah,
your tank. So if it works in the
absolutely.
ocean, I figured, well, it's got to work in a small body of water like your tank.
Yeah, for sure. And so this is the website right here that people can go in and order a sample. And then from here, so once you order the sample, do your test, you have to register it,
Yep.
and then you send it back, and then you get your results. And so I guess we can go ahead and jump into the results right now from the
Yeah,
tank.
let's do it.
So I'll pull up the microbiome report here.
Great.
And so the first thing that we see once we get it back is a diversity score. So what does that mean? I mean, I know green is typically good, so you probably want it in the
Sure.
green. This one's a little low. So what does that mean for
Yeah.
the tank?
Well, let's start by acknowledging that the color code there is sort of based on our recommendations, our assumptions. We consider high diversity to be good, but really let's focus on the numbers. The data themselves. So diversity, simply the simplest way to understand this, it's just the number of different types of microbial DNA that we found in your sample. There's a bit more that goes into it. There's some corrections for differences in the total number of DNA sequences, but it's basically how many different types of DNA did we find in your sample. And you can see that we compare that number, your diversity score, with all the other tanks in our database. We have this huge database at this point of all the healthy reef tanks that we've studied, and we compare your sample with all of them to ask, how does it compare? And you can see that yours was what is that, the 6th or 8th percentile? Very low, right? So it was, the majority of reef tanks that we've sampled had a higher diversity score than yours did.
Okay.
Now there's a lot of debate about how much does diversity matter? Why does diversity matter? Even if diversity is valuable, how much is enough? How much do you need? These are questions that aren't completely settled among professional ecologists. They're certainly not settled among reef keepers. I think there's room for discussion and debate here. I think about this in some ways like alkalinity. We can tell you how to measure it, but everybody has their own opinion on what is the best value that they want to maintain their tank at. And I think there's, reasonable people can reach different conclusions there. My own conclusion is this: in nature, I mean, there's over a century of study in ecology of diversity. And in nature, diversity is generally associated with a more productive ecosystem and a more stable ecosystem, a resilient ecosystem. When it gets disrupted, it bounces back rather than crashing. These are all things associated in nature with higher diversity. And ecologists have spent a century arguing about why. What's the mechanism? How does diversity help? I sure don't have the answer to that question. The way I think about it is one part of the value of diversity. It's a strategy for catching the bacteria that you want. Let's imagine that there's some bacteria you want in your tank and some you don't. Well, if you catch very few bacteria, you probably haven't got all the ones you want, right? By catching more and more bacteria, by establishing a more diverse starting set of bacteria in your tank, you have a better chance of catching the ones that you want. Now, having said that, I have to acknowledge the obvious counterpoint that, well, you could catch the ones you don't want to. And I mean, I think that's where testing comes in. Let's not just grab water from every pet store tank in the hopes of maximizing diversity. Let's think carefully about what we're putting in the tank. But yeah, those are my thoughts on diversity. It's generally a positive thing in nature. I think it is also a positive thing in our tanks. Now let me take a little opinion out of that. I said I think it's a positive thing, but I think we've all observed that as as a tank gets more mature, corals do better. So in other words, when you first start out your brand new clean reef tank, and you try to put a coral in there, it generally doesn't do well. You know, dry sand, dry rock, plop a coral in, and it's not going to do well. We've generally in the hobby observed that over time, something happens to the tank as it gets older, as it matures, that makes it work better for corals, provide a better habitat for corals. We didn't know what that was. There was something we weren't measuring. But we've shown clearly in multiple experiments, and other people have shown it too, that over time, what happens in a tank, you first establish a tank, it has very low diversity. And that diversity gets established. It increases over time. If you start with live rock, you can get to a good diversity score within a month or so after starting a new tank. But in a new tank, diversity is low. And I think that's a good piece of evidence that that diversity is beneficial, because as diversity increases, the tank becomes more able to support corals.
Yeah, and I think a lot of times, either being new to the hobby or first setting up a tank, sometimes we might think that, oh, it's a water chemistry issue that our corals aren't doing well, when really our microbiome hasn't been established enough, or like you said, there's not enough diversity in there for the different functions that they do to help your reef tank. And so the next thing after diversity that you look at is the balance score. And so
and yours scored very high on balance.
So this one.
So balance, this is a measure of how similar is the microbial community in your tank to the typical microbiome in an established healthy reef tank. So we're taking these two complex communities, comparing them, and giving you one number, the balance score. That number is going to range from 0 if it's really, really different, all the way up to 1 if it's completely identical to the typical reef tank. We take that score and again compare it to all the other real tanks that we've tested, and ask, how does yours compare? You can see that yours was in the 92nd percentile. So you had a higher score than 92% of the tanks we've tested.
Very cool. This so it's pretty good.
Very typical community. It's very similar to the mature reef tank community. This is a very positive sign. This is something that takes time to develop. You know, you can rapidly achieve diversity by sort of putting a bunch of stuff in your tank quickly, like live sand, live mud. You can quickly get diversity. But this balance score, this always takes time to develop. This is really a measure of how mature the community is. Over time, we'll see one family rise to dominance and then kind of collapse, and another family takes over. That process takes some time. And it's a lot like the same succession process that happens on land in terrestrial communities. If you've ever seen a field turn over the course of years from a field into a bunch of shrubs and then eventually into a forest, that forest is the long-term stable ecosystem. And the microbial community in your reef tank goes through a similar process. It's just invisible. You go through different stages as that community matures. And what we're seeing in your tank with this very high score is a tank that looks a lot like that mature long-term community, sort of the community analogous to an old growth forest on land.
Very cool. And that's probably due to having my 5-year-old tank and then moving everything over to start this reef tank.
Absolutely. Yeah.
Very cool.
Yeah, I think that's, you know, as long as the tank's healthy to begin with, right, not full of parasites, that's a great way. Why throw away all that progress you've made, you know, building that community over years.
Yeah, very, very cool. And then from there, you have the actual community composition. And this is the makeup of the tank, all the different bacteria that's living within your tank.
That's right. Yeah. Yeah. And this, this picture, this really focuses on the water sample and compares it to the typical profile in other water samples. And, looking at it, I think visually your sample is just kind of obviously very similar, right? We can find all the major families from the typical sample. You can find them in your sample generally at a pretty similar level. One that I always look for in particular is that pink one, the Pelagibacteriaceae. This is a group, one of the first members of the group studied was called SAR11, SAR11. Kind of wish they'd stuck with that name. It's easier to pronounce than Pelagibacteriaceae. Yeah. But in any case, this group, it's the most abundant group on natural coral reefs. It's well adapted for low nutrient conditions, and it's a true bacteria plankton. It floats around in the water, free swimming in the water.
Okay.
That group, it takes time to develop in the tank. We never see that in a young tank, in a brand new tank. It always takes time to get established. So I consider it a good marker, A, of appropriate nutrient conditions, because it likes pretty low nutrients, and B, a good marker of maturity of the community.
So because it's free swimming in the water column, this is probably something that gets taken out by like UV sterilizers quite a bit.
Exactly right. Yeah, that's the group that if you have a UV sterilizer, we generally find 0 of this group in your tank. That confused me a lot when I first started sequencing tanks and seeing that variation. But once we got enough data, and I was going through every now and then I go through the database and ask questions like this. Mm-hmm. And it just popped out that it was such a strong signal. It's something like a 20-fold difference in the levels of that family. I mean, it's huge. And really, it's like most tanks that have UV just don't have any of that tank. It's an absence.
Wow.
And most tanks that don't have UV have that group unless they're very young, or there's something else wrong with them, right? Yeah, so that's a good example of the kind of thing that we're learning over time at Aquabiomics, really in collaboration with the reefing community, because I didn't set up all those tanks with and without UV sterilizers. Mm-hmm. You guys did, right? Yeah. And it was by having the community do that experiment and run tests with us that we were able to see that difference. And it's fun. I mean, we've actually been able to use that. I have a client, at least one client, who's experimented by turning his UV on and off and seen it appear and disappear.
Oh, very interesting.
In response. Yeah. So it's a good example of, if we think about the microbiome test report, or the microbiome as having sort of a bunch of dials and a bunch of knobs on it, right? The dials are the indicators, the data that I'm giving you in the report. We would like to have a lot of knobs we could turn in response, where we could mm-hmm. say, oh, I have too much of that group, so let me turn this knob and dial it back down. Or I don't have enough of that group, let me turn this knob and dial it back up.
Right.
And that's what we're working towards. This is one of the examples of a very clear knob that the user can turn. If you turn on your UV, that pink bar will go away. And if you turn off your UV and add some water that contains that group, it will come back and recolonize your tank.
So these are most common in like a successful reef tank or a nice healthy reef tank. So these are obviously more of the good bacteria. What's some of the bad bacteria that you could get?
Yeah, and we can sometimes get a clue at that right at the stage where we're looking at the community composition. This picture here, the kind of hot pink group at the bottom there. This family is called Vibrionaceae, and the beginning of that word should sound familiar. Vibrio. I think most people in the hobby have heard of Vibrio. This is a genus that includes a whole lot of pathogens. Fish pathogens, coral pathogens, and there's even some human pathogens in there. So it's a nasty group. We sometimes see tanks that have an elevated level at the family level, so there's a big hot pink bar in this graph, and right away we can say, oh, look, there's there's likely to be pathogens. You have so much Vibrio. There's a couple other groups of pathogens that sometimes show up here too, but of course, the real diagnosis of pathogens happens at the specific sequence level, not the family level. So we leave that towards the end of the report if you want to scroll down.
Okay.
I think you might have had some pathogens. I think you had one fish pathogen and one suspected coral pathogen, right?
Yeah, I believe so. And this talks about your nitrifying community. So
Sure, we
you
can.
want to touch on this real quick?
Yeah. Let's
Okay.
let's talk about this. So the nitrifying community, this is a community that the reefing hobby, we knew about this, right? Before we ever came along at Aquabiomics, the reefing hobby already knew about the nitrifying community. It was probably what we knew most about. And because there were publications on microbiology out there, we could kind of put names to the bacteria that must be doing this. But of course, we had no way of knowing which bacteria were doing it in your tank, and were they there in your tank yet or not. All we could see was the chemical effects. So this was one of the motivations for doing this testing in the first place. We can break this group, nitrifying microbes, down into two areas: the ammonia oxidizers and the nitrite oxidizers. One of the things that we've learned from this surveying of hundreds and hundreds of tanks is that ammonia oxidizers are consistently they're present at higher levels than the nitrite oxidizers. They're more abundant in the reef tank. And your sample, fit this pattern exactly. So we saw more of this group than the other. And it looks like we actually had pretty high levels in your towards the upper end of the normal range, I guess. So the typical range over on the right-hand side describes between, I don't know, I think the 10th and 90th percentiles, something like that. And so yours is towards the upper end of that range. So normal on the high end, very positive sign. One other thing I'll point out while we're looking at this table is this group of microbes, these are actually not bacteria at all. These are archaea. And so people may sometimes wonder, why does he keep insisting on calling them microbes, not bacteria? Well, look, one of the most important bugs in your aquarium is not a bacteria. It's archaea. And that's actually the rule across the hobby. The dominant ammonia oxidizers are not bacteria. They are archaea in this group. So the ammonia oxidizing part of the community looked very typical and good in your sample. The nitrite oxidizers, I think, were low. Is that right? We did not detect any in this sample. Now, these are surely present somewhere in an established tank. But what these data indicate is that in your biofilm community and in your water community too, they're present at very low levels. Actually so low we didn't even detect them in this sample. I was looking back at your first test. Remember, we tested a sample from your tank previously, and the water filter part of it didn't work, so we didn't have a good report. But the swab sample actually worked fine that first time, and we did find nitrite oxidizers that first time at very low levels. So, really, I think the two tests are telling us the same thing. Nitrite oxidizers are at low levels in your system. Now, is that a problem or not? I always tell people it's only a problem if you're having issues controlling your nutrients or nuisance algae, because there are other processes in the tank competing with nitrification for these nutrients. And so if we find low levels of these groups, it probably means they're not getting enough nutrients to, rise to high levels. That could be because of carbon dosing. I don't recall if you do carbon dosing. It could be because of macroalgal refugium or an algal skimmer. Do you have anything like that?
Yep, I do run a refugium, so there's some chaetomorpha down there.
Yeah, that's great. And I do too. It's not a criticism of those at all. It's just a discussion of kind of the interaction between different pieces of the system, right? If you have two parts of the system competing for the same nutrient, every time you turn on your light in your refugium and take up some nutrients out of the water, those are nutrients that the nitrifiers won't get. And so these communities can be lower as a result. Sounds to me like it's not at all a problem in your system. Just something to be aware of. I run a refugium, and my light bulb burned out recently. I just use a cheap, grow light.
Yep, same here. Yeah.
The light bulb burned out. I started getting more algae than usual on my glass in the display. And it's an anecdote. I mean, I'm just telling a story and waving my hands, but I think it illustrates the process that I'm describing, this interaction where if you don't have a high nitrifying community, when you turn off the light in your refugium, there's nobody else to take up the nutrients, or there's not enough of them, right? You can become dependent on whatever process you're using. And I think that's the case for any of those three processes, right? Whether it's carbon dosing or nitrification or macroalgal growth, the tank will become dependent on that process. And if you suddenly remove it, it would take some time for the other processes to grow to the point where they could take it out.
Yeah. Okay. Very interesting. So that's one. That is the reason why they say, like, don't just take all of your refugium away all at once. You want to harvest it out slowly, and so that process stays going.
I definitely agree with that.
Yeah.
And part of the lesson that we all learn in the hobby, right? That nothing good happens quickly. If you're making changes, make them gradually and slowly, kind of wean your tank off of it. Yeah. So if I remember, you had one of the most common fish pathogens in the hobby, Vibrio fortis.
Mm-hmm.
I think that was the one.
Yeah, I think so. I know. Popping this back up on the screen here. Like I mentioned, I had cyanobacteria, but it looks like it was pretty well taken care of. It looks like there's a few different groups still in here. If you want to.
Yeah, so I see one group here, and it's at pretty low levels. It is a group that includes some filamentous types. This is one of the things I'm always curious about. What kind of cyano do you have? Some are filamentous. That is, they form filaments. Those filaments form the mat, that kind of ugly. Yeah, it's called a mat.
Yeah.
A bacterial mat.
Similar to this right here?
Yeah, I mean, it's similar to really all the nuisance algae. They all grow that kind of mat down on the sand, right?
Mm-hmm.
Well, the filamentous types of the cyanobacteria are the ones that form mats like that. And so that's a question I always ask. Your sample did have a filamentous group of cyano, but it was at very low levels.
Right. Oh, that was dinos that I just showed right there. But yeah. But yeah, so very low levels of cyano. That one popped up. That was one of the first things that popped up when I set up this tank. Was cyano, and then I knocked that out with like a red slime remover.
Got it.
So that probably affected the biome a little bit.
Sure. Yeah.
But yeah, it was pretty effective. But yeah, so fish pathogens.
Yeah, great.
Vibrio fortis, yes.
Vibrio fortis, yeah. So super common in the hobby. Something like half the tanks that we've tested had this. I mean, it's really common, but probably not a concern in most tanks. The only papers I can find really linking it to disease are in seahorses and their relatives. So, before you put that expensive pipefish, what's the pretty one, the blue striped one or whatever, before you put an expensive pipefish in here, I would want to check if you have it. But most of us aren't raising pipefish and seahorses, so it's probably not a big issue. With that said, it is a pathogen that in direct experiments seems to affect a wider range of fish. Like if you deliberately infect fish with it, it can affect a wider range. So it may be contributing to some issues in the hobby. I don't think it's really a big issue to be worried about unless you're growing seahorses, because it's in so many tanks. Mm-hmm. If it was killing a lot of fish, we'd know about it by now.
Gotcha.
So I generally pass over Vibrio fortis and say not a big concern. The coral pathogen is an interesting one. So I'm calling it a suspected pathogen because there's a formal process that scientists go through to designate a bacterium as a pathogen. It involves sort of 5 or 6 major experimental steps, and they're really hard to do in corals. We're certainly not there for this disease. The disease is called Sctld, Stony Coral Tissue Loss Disease.
Okay.
Unfortunate acronym that's hard to pronounce.
Yeah.
It's devastating corals in the Caribbean right now. The last few years, it's been a huge problem. It's a major ongoing area of research. And every time one of those papers comes out, I grab the DNA sequences from it and compare it to our database and say, okay, do we have any of these bugs in the hobby? And it turns out that some of them are pretty common in the hobby. So the paper I pulled this sequence from, they identified something like 7 or so microbes that are associated with the disease. And I keep saying associated. So what I mean there is, like, they find this bacterium in the diseased corals, mm-hmm. either more frequently or at a higher level than in healthy corals. Some of these bacteria associated with the disease are really strongly associated. Like, you only find them in the diseased corals, and you always find them in the diseased corals. So it's pretty good evidence. It's just not enough evidence to conclusively say that the bacterium is causing the disease. Okay, so they came from peer-reviewed publications from this ongoing research, and I found the exact same DNA sequence in your tank. This was for one of the common ones. I don't recall. Let me pull up the report on my screen so I can see it better. The Planktotilia species. So this is one of the more common ones. This guy occurs in 19% of the tanks that we've tested. So, about 1 in 5 tanks has it. The levels in your tank were not unusually high. They were within the typical range. So your tank has a suspected coral pathogen. It's absolutely there. It's the same one that's present in about 20% of tanks, and the levels in your tank are similar to what we see in those other tanks. Okay. So what do we do with that information? Well, if you have ongoing coral diseases in your tank, I think this is a prime candidate to consider. We haven't found any other coral pathogens or any other coral parasites or pests in your samples. This is the only coral bug that we've found. So I think it's a good candidate. That doesn't mean that I'd recommend you dump a bunch of antibiotic in your tank or something like that.
So is a solution maybe to pull those corals out and do a coral dip, or since the pathogen is in the tank, that's really not going to affect the corals, because like you were saying, it's like if the coral's healthy at this low level, the coral may be okay. But it's when the coral becomes unhealthy that it starts just taking it down.
Yeah. So, I think the advice always comes down to a case-by-case basis. Now we know the bugs in your tank. Mm-hmm. So if we see corals doing badly, and it sounds like you still have 1 or more corals showing symptoms, is that right?
Yeah, it's mostly euphyllias, hammer corals. Yeah. I've had a trachea go down too. So
yeah. So then there's a couple things that we could do at this point. You could start experimenting with the benefits of an antibiotic dip. The bugs in your tank, mm-hmm. but that doesn't necessarily mean that you have a colony of it, a population of it colonizing the corals themselves, right? So you could take kind of an experimental approach to explore that, saying, well, if I take one of my euphyllia out and do an antibiotic dip, do I see any improvement? If you do see some improvement, that would be consistent with the idea that this bug, we now know it's there, is contributing to the disease, and it might support kind of moving forward and treating the rest of them. If you do that, my recommendation would be amoxicillin. It's not the easiest one to find, but it is out there, marketed for fish. And I would use it at the similar concentrations that they recommend for fish.
Okay. It's probably a really small dose.
Yeah. So, yeah. I mean, that's, I'm not a fan of the sledgehammer approach with antibiotics, because we know there are such a thing as beneficial microbes, even if we don't know what they all are, and even if we don't know what they're all doing, we know there's such a thing as beneficial microbes. So I try to start on the low end with any antibiotic treatments, whether it's a dip or an in-tank treatment. Let's start on the low end, and see if we can kill the target without killing everything else. So yeah, some low dosage dip of amoxicillin is something you could experiment with. That's a path you could take. Another path we could take at this point is to say, okay, I know it's in the tank, but I don't know, is it actually infecting my corals? And we could follow up with a swab sample of an infected coral itself, to pursue that. That would be, I guess, the more cautious way. We could take it step by step and say, I'm going to first check, is it actually there? So that's an option too.
Okay.
Could take a wait and see approach and just say, well, if there's not any major outbreak, maybe I just don't touch anything and see what happens. But just this knowledge that there's a bacterial pathogen in the tank, kind of keep that in your pocket. And if you see a major outbreak emerge, you don't have to do a lot of research all of a sudden to figure out what is it. You can act quickly and say, I'm going to do an antibiotic treatment because I know I've got a bug.
Yeah, because I've been really slow to add coral. Like, I love hammer corals and frog spawn corals, but I've been really slow to add them only because I see them decline.
Yeah.
And in previous tanks of mine, it really hasn't been a problem. So I'm mainly adding like mushroom corals and zoanthid corals at the moment.
Yeah.
So.
Yeah, it's tragic when Euphyllia die because they die so quickly, and they're pretty, and they're usually not cheap. Yeah. Well, since you're a fan of Euphyllia, I'm glad to see that we didn't find any of the Archibacter. So there's another one of the really new things that we've learned in our work at Aquabiomics is this pathogen that causes. I'm convinced it is the cause of brown jelly disease. Now that there may be other contributing factors, the work is not done, but I believe it is the cause of brown jelly disease. We've seen this association so strongly, repeatedly. We find the same thing in tanks from Europe, down to the identical DNA sequence, not a base pair different, associated with brown jelly disease. So it's really nice to not see that in your tank. We saw this suspected coral pathogen, and I'm kind of giving you this soft advice about, well, I wouldn't nuke your tank over the presence of this. You might move slowly, right?
Yeah.
If we saw Archibacter type 1103 in your tank, I would feel differently. It is one that I don't like to see at all in a tank. I don't consider there to be a safe level, especially in a tank with Euphyllia. Yeah.
Very interesting. Yeah. So get that one out as quick as possible.
Yeah. Now, so we didn't find the worst ones in your tank. I guess that's the context I'm trying to share.
Well, that's good. That's very good. Now, is this can you kind of wait this out? Is this the type of thing where, like, if other archaea or bacteria is like out competing this pathogen, does it disappear over time? Or is it just hangs around doing its thing?
Yeah, I mean, I think the safe answer here is to say that we don't know yet, but I'm going to speculate based on what we know that I think that this group of bacteria, we should expect them to persist in the aquarium even without any corals around. This is not like it's not like leaving the tank fallow to get rid of ick, that kind of thing.
Yeah.
These are normal parts of the community. So these bacteria associated with Sctld, they're all part of the Rhodobacteriaceae. This is one of the top 5 most abundant groups in the microbiome. It's the dominant part of the biofilm. That is the family Rhodobacteriaceae, dominant part of the biofilm. And these pathogens or suspected pathogens, they're living right in there with all the other Rhodobacteriaceae in the biofilm all over the tank. They're a normal part of the community, not just around when there's an infection. So
Yeah.
I am skeptical that, short of an antibiotic treatment or something, I'm skeptical that we're going to wipe them out of the tank. We recently, of course, can't give any names, but we recently sampled a bunch of tanks from a fish dealer. Somebody who buys and sells lots of fish. They don't deal with corals at all. Not a coral in their facility. But boy, every single one of their tanks had these Sctld pathogens in them. So they're definitely growing in tanks, even when there's no corals around. They're widespread in the hobby. I'm skeptical you would get rid of it by waiting it out or changing conditions. Yeah.
Yeah. Very interesting. Yeah. I mean, we get corals from all over the place, many different stores. So we're bringing in a bunch of different water. I mean, you acclimate corals and you try not to add that water to your tank as like a rule of
Sure.
thumb. But the tiniest drop
Yeah.
can,
Yeah.
yeah.
And these guys, these members of the Rhodobacteriaceae, any of the Sctld associated bacteria, they're biofilm, right? So they're clinging to the coral frag plug, the coral skeleton. They may actually be whether it's healthy or diseased, they can be a part of the coral microbial community itself on the coral tissue. So,
Gotcha. So not just the water; it's on the plug as you're putting it in the tank.
In fact, primarily not
Yeah.
in the water. It's primarily on the plug. Yeah, we find it in the water too, but they're mostly biofilm organisms. So,
Gotcha. Yep.
yeah, these things are going to get into your tank. It is likely, but not inevitable. We don't find them in every tank, right? This one is only 20%, and it's one of the most common ones. The other members of the group, they're rarer. They show up in maybe 5% of tanks. So I am a fan. I do think all of this testing, supports the practice that many of us have adopted of being really careful about what we put in our tanks, right? Not necessarily nuking it with harsh chemical dips, but at the very least, cleaning it well, inspecting it well, putting in a minimum amount of external material and making that material as known as you possibly can, right? Inspected or treated.
Yeah, and with these tests, one of the goals, I believe, is to have a little bit quicker results. Is that something that you're working on?
Yeah. Let's talk about that, kind of the development of the testing service. Yeah. So the point I always like to start here is, in the academic world where I come from, these tests are done on a time scale measured in months to a year. Usually, when a researcher hands off their sample to someone, they're happy to get results back in about a year, sort of for their big conference next year or whatever. I knew that wasn't going to cut it as a service, right? So I've made a lot of efforts to really make this go as quickly as we can. But the turnaround time has always been the biggest challenge. Hobbyists want the results tomorrow, and the facilities that do this kind of work are expecting to give you results back a year from now. What we have done in phase 1, that is Aquabiomics, up until about a few months ago, we were doing 1 month turnaround time. Every month we processed a batch of samples. And so depending when your sample got to us, your turnaround time could be less than a month or around a month. And the reason for that, it's not just that we're lazy or limited by resources or something. It's the technology. The DNA sequencing machines, you can't just put one person's sample on there. You need to put enough samples on there to make use of all the data that you're going to get back. And so, that was the limitation. It was just about, we need to get enough samples to fill the batch. And in the early days of Aquabiomics, the first couple of years, that meant one batch a month. A batch being maybe 50 or so 50 to 100 samples, depending what kind. Over the last few months, say half a year or so, boy, we've been so busy. We're now running 2 batches a month consistently. And so this is without any changes in technology. We're just working more, just working more hours. We're now running 2 batches a month. So again, depending when your sample gets to us, you could get your results less than that, but generally it's going to be around a 2-week turnaround time.
Yeah.
So I think clients should really, keep that in mind in terms of using the service. It's a good idea to have your test on your shelf already before the problem happens. Because, when you place your order, let's say you have a sick fish, you have an algae outbreak, whatever the problem is, when you place your order, well, you've got to allow some time for us to ship it out to you and the postal service to get it to you, right? So there's some time passed. You would save that time if you had the testing kit already on your shelf. So that's one way to minimize turnaround time. Another thing you can do, depending on the nature of your sample, whether it's an emergency or not, you could think about our testing schedule. So these days, what we're doing is the end of the month and the middle of the month.
Okay.
Those are our sample deadlines. So, if you get your sample to us right at the middle of the month, say like the 14th or so, that would be right before we begin that batch. Do you see what I mean?
Yeah.
So you would have the shorter turnaround time. And again, you'd have another sweet spot at the end of the month. Get it to us right before the end of the month, and we'll start processing it sort of right away.
Yeah, so definitely be prepared, have the kit, and then know the timing of the schedule.
Right.
Yeah.
Yeah. So that exactly it. Those 2 things are the best things the user can do to minimize their turnaround time. And the other thing that you can do is just be aware of this schedule. So plan ahead. Don't try to use the service as something to get an answer tomorrow. But things don't always happen quickly in reefing anyway, right?
Yeah.
You get your fish, you put it in a quarantine tank. You're probably going to leave it in that quarantine tank for weeks to perhaps a month. Well, that's the kind of time frame of our testing, right? So I think if the user just thinks about it that way, as I'm going to test it and then wait a month, which is what we have to do with a lot of things in reefing.
Yeah.
If you go into it with that expectation, you will be happy, because we will be able to get you your results within that time frame. But
because
overnight results aren't going to happen anytime soon.
Yeah, because in some cases you have like a fish breaks out with something like at that point you're able to see what it is. If it's like cryptocarin or like ick, you can see that. You can make the judgment. You don't really need a test to tell you that you have it at that point. So this is kind of like, you want to send in your test to see if you potentially might have something, and then you can proactively, like, you can get on it before it goes south.
Yeah, that's right. Yeah, so it's all about planning ahead and taking the samples in a time frame that's realistic for the testing. So thinking about the fish example, imagine you got in a new fish and you put it in your quarantine, and you don't see any symptoms, right? You don't know, is that thing infected or not? Well, go ahead and test it. Test your quarantine at that point. After the fish has been in there a couple of days, go ahead and test your quarantine. And then when we give you your results back and tell you that tank was clean, maybe you don't need to wait so long before you put it in your tank, in your display. Do you see what I mean? You could use it. You could use it in conjunction with a quarantine to evaluate whether there is an unknown disease. I think it's equally important to test the tank. So, a lot of us pay a lot of attention to the fish we're putting in our tanks, because we're aware that, if I put this fish in and it introduces ick into my tank, it could wipe out my tank. But there's been less discussion of the opposite. That is, there's already stuff in your tank that could pose a threat to that new fish. And so it's useful to test the tank before you put the new fish in. And that we have a lot more flexibility on the time, right? test your tank now, and once you get the all clear result, go ahead and buy that expensive fish that you've been wanting to buy.
Yeah, so like in my case, I can add a fish to the tank. I just probably don't want to add a seahorse or something to the tank.
Yeah, that's right.
You
I
know?
mean, take the information you got and make the right decisions with it. There are other pathogens and parasites that if I saw them in your tank, I would say just don't buy any fish for this tank, right? We get people back with such high levels of ick in their sample. I certainly at that point would not add any fish to the tank.
Right, yeah.
But that's a very different situation than what we found in your tank. Yeah.
Yeah, very cool. So do you want to jump into the tank DNA test report? And
Sure, yeah.
And there's some interesting things that popped up in there. So let me add that here.
Yeah, great.
So this is...
Yeah, so again, it's DNA sequencing, but here we're just sequencing DNA from different organisms in the tank. We've targeted the eukaryotes, and that's really everybody that's not prokaryotes. So if it's not bacteria or archaea, then it's in this report. And that's not an artificial distinction in terms of, well, we just split it up. I mean, we do an entirely different process in the lab in order to get the two different kinds of data. So one of the groups of eukaryotes that we can find here that we're interested in are the nuisance algae, like dinoflagellates and diatoms. And so this method of testing can tell you not just, yes, you have diatoms, yes, you have dinoflagellates. It tells you how much and which kinds. So based on the DNA levels, we found low levels of dinoflagellates in your sample. They were amphidinium. That's one of the well-known groups of dinoflagellates that lots of people discuss in the hobby. And, there are different treatment options discussed depending on what kinds of dinoflagellates you have. So it can be useful to know which type. In your case, there wasn't a lot in the sample, and it sounds like you had already kind of defeated the dinoflagellate problem by that time.
Yeah, I was on my way out of getting rid of them for sure.
Yeah, cool.
And so that definitely kind of answered for me what type I found in the reef tank.
Yeah,
I was
it's great
comparing
having
off Google, and here it is in the results.
It's great having those two lines of evidence, the visual confirmation and the DNA. At that point, it's hard to debate was that it.
Yeah.
We also sometimes find toxic dinoflagellates or parasitic dinoflagellates. Some of the fish parasites show up in this group, and there are also sort of toxic algal bloom types. None of those showed up in yours, just a little bit of amphidinium. If we scroll down, I mean, we saw diatoms, but there were no unusual types, just high levels. In this sample, we found high levels of diatom DNA from one type. Very common type. I wouldn't be concerned unless you are seeing an outbreak, in which case you might take this information to say, maybe I have a silica issue, or maybe I just need to do some sand vacuuming.
Yeah, I think I need to do a little more cleaning up on the sand and the rock work for sure.
It's funny. I mean, I spend all this time working with high-powered computers and sequencing DNA, and often the advice I end up giving people or the suggestions I end up giving people are so dumb. It's like, well, try vacuuming your sand.
Yeah.
So I shouldn't say dumb, but such a low tech,
and a lot of times that gets overlooked, right? Like in the busyness of life, just day to day, like, okay, I should probably vacuum the sand.
There's no substitute for physical cleaning.
Yes,
manual removal.
Yep.
So we can scroll down and look through all the other stuff we found. So it really does just find all of the eukaryotic DNA in your tank. And so looking at this list, I can see that we have fish in this Actinopterygiae group. We have fish. We have sponges. I see snails, copepods, worms, and then it kind of goes off the screen, so we can't see more. But the point, just while we're looking at this, is to say we see a wide range of animals. I just named a few that are consistent with what you'd expect to find in a saltwater aquarium community.
Yeah, and you had sent me a message that you found like an odd, I think it was the gastropod or something that like that. You found a DNA that you don't normally see, and you were kind of wondering what that was.
I remember that the snail in your sample was an unusual one. Let me pull that up and see if that's the. Yeah, I was just looking at your result again today, and I didn't spot any unusual parasites or anything, which is always good. We do sometimes. Oh yeah, here it is. So actually, it's a nudibranch. We found a nudibranch.
Oh, okay.
So yes, you remembered correctly. It was gastropod. It's not a snail. It's a nudibranch. So I'm not at all confident about the species ID on that nudibranch.
Okay.
But the higher level, family level classification, I would stand by. So it does seem to be indicating the presence of some kind of nudibranch. It is not one of the known parasitic nudibranchs.
That's good.
There's only a couple of them that we know. It's possible there are others out there.
Yeah.
So what I'm going to tell you is that is a nudibranch. It is not one of the known bad ones, but I'm not entirely confident what kind it is. Okay. I can tell you what the DNA matched most closely to, but it could be a relative of that one.
Very cool.
Yeah, interesting. That's an oddball. So you don't know about the presence of any nudibranchs in your tank?
I mean, not that I've seen. No.
Yeah, they can be cryptic. We can find stuff. Certainly, a lot of the stuff on this list is invisible, right? You probably couldn't have told me about all the different ciliates that were in your tank either, for the same reason. They're just hard to see. Yeah, that is an interesting finding.
Yeah, and then so what's the okay? So this is diversity and
right. So the first graph shows you the data in terms of diversity, and the second one shows you it in terms of abundance.
Right.
So in the second one, when you see a big bar, that means there's a lot of it.
Okay.
In the first one, if you saw a big bar, it meant that there were a lot of different kinds of it. Two different ways of evaluating sort of what was there a lot of in your sample. And yeah, it looks like in your sample, yeah, forgive me, in the one on the screen, it's hard for me to see, but if I go to the one on my screen, I'll be able to see the colors better. So it looks like there were two big dominant groups in your sample, and I'll scroll down to. So one of them was the Phaeophyceae. So this is a group of macroalgae. Okay. And the other one. The other one is diatoms. So the two big abundant groups in your sample are diatoms. We already knew that's the green one.
Yeah. The
big green one, and then the big blue one is Phaeophyceae, a group of macroalgae.
Okay.
Yeah, so that abundance information, I would not interpret that in terms of sort of counting organisms in the tank. This is different than the microbiome, because think about in the microbiome, everybody has just a single cell, right? They're all unicellular organisms floating around. So you can somewhat count the DNA and infer how much of it is in the community. There's a lot of arguments to be had about those details among microbiologists. But in the eDNA world, it's very different, because we have multicellular organisms. You have things like a big sponge with billions of cells in it, right? You're going to get a lot of DNA from that thing. And then you have other things that are very tiny, and you'll get less DNA from them. So I wouldn't interpret this abundance in terms of the abundance of the organism. Really, it's just about the sample. It's just saying that we got a lot of DNA from algae, macroalgae, and diatoms in this sample. Yeah. I think one of the most interesting things in these reports can be when we scroll down and look at the complete table at the bottom. So I have so much fun. Every batch of samples I process, this is my favorite part, is when I get to see the tank DNA reports for the first time, and get to look through all the oddball stuff that we find in people's tanks. So as you're scrolling through, I'm seeing Dinoflagellates. So that's a obscure little group of microscopic eukaryotes. Diatoms, we all know about diatoms. Um, Here's that oddball gastropod. You see it's in the family Heterobranchia. So these family level designations are going to be very accurate. It's certainly some kind of a heterobranch. The macronucleata. This group is the ciliates. We know these commonly as ciliates. That's their formal taxonomic name. And so this is a group that's important to look at because it has lots of parasites in it, right? That is the group. The group includes lots of parasites. We found lots of ciliates in your sample, both in terms of the abundance and the diversity.
Okay.
I don't know, a dozen or so. What is that, 8 or 10 maybe? They're all very common types of ciliates, but none of them are the parasitic kind.
Okay.
This is something I like to look at in the reports. I like to say, look, we sampled the ciliate community effectively. We got good data on the ciliate community, but we didn't find any of the parasitic types.
Yeah.
If I got data back and we just didn't find any ciliates in it, I'd be a little skeptical about claiming that there were no parasitic ciliates. So that's one reason I like to look through these tables, is to kind of verify that, yeah, we sampled this tank really thoroughly and still didn't find any parasites.
Yeah, no, it's I'm very fascinated with all the stuff that you're able to pull out of this water sample. So
yeah, I mean, it's great fun. You really can find stuff you weren't even planning on. So, you know, here's copepods, the maxillipod group. So that's a very common kind of copepods in aquarium samples, not a parasitic type, not any kind of problem. But, you know, there are some parasitic types of coral copepods. So that's certainly something I look at every time I get to the copepod section and say, well, are there any oddball parasites in there? No, this looks clean.
Nice. And so, and with the reports, you also have the families and different things linked up, right? So that you get more information.
Yeah. So this is something I'm constantly working on, that as time goes by, this part of it will improve. That's in part because I will have the time to do more work on it, and in part because we will learn more about things as we go along. So my default starting point with these is always just, I wrote some code that says, well, if there exists a Wikipedia page on this, link to the Wikipedia page on it, because that's certainly going to be a good starting point to learn something about. But as I learn more about these groups and build pages for each of the specific groups, I'll have the code instead link you to the page on our website. So so I'd encourage people to always check these links. This really is a good, you know, I'm intending to give the client all the information I can about each of these groups, and the way we're trying to do that is through the links rather than typing it out in an email.
Yeah, I think that's a huge benefit, because then that helps us understand exactly what we're looking at if we're not familiar or we haven't jumped into this micro world, and so we don't know all the terms and everything, so we can see that.
And look, I taught invertebrate biology for a long time, but I don't know all these names, right? So I mean, when I pull these lists up, it's very frequent that I see something, I don't know what that is, and I click the link and go down the rabbit hole learning about it. So so I'm that's high on my priority list to add more content to these links so that they can go to specialized pages on our website and give you more details, sort of more focused for the aquarium hobby.
Yeah, very, very cool. And so if people want to get their own test, then they go to the website, which I have a link right here. So that's aquabiomics.com.
That's it.
And then you're also doing like live reef sand, and you're doing the rock rubble as well. And the goal with that is to help seed the tank and get you on the right track. Okay.
Yeah. Yeah, it's something, again, I think about this in terms of sort of dials and knobs. We test your community, you learn something about it, but then you want to turn a knob and adjust it. You want to change something about the community. So in the long term, my long-term plans for the company is that we will continue expanding on these things. We're very focused on testing right now, but long term, we want to expand on these offerings of products that you can add to the aquarium. Right now, there are so many of those products out there that it doesn't seem like the right strategy for us to make that priority number one of being the newest bacterial supplement on the block. But what I didn't see a lot of on the market, I couldn't find any good source of live reef rubble on the market, live reef rock. So when I say rubble, what I mean is really just tiny little pieces of live rock. We're literally breaking it up and homogenizing it so that every cup is sort of similar to every other cup. So it is just live rock. When I started the company and I did an experiment on live rock, demonstrating yet again what we all knew in the hobby, that live rock was a really effective way of seeding a diverse and typical reef tank community. I just couldn't find good sources of it on the market. There's all this crap that's, purple dyed cement with some stuff growing on the outside of it.
Yeah.
It's not real live rock, So we found a supplier and started selling this live reef rubble, which is just broken up live rock. The most important thing in my mind about our products, the live reef sand and the live reef rubble, the most important thing is the fact that it's tested, that it's known to be parasite free. It's really not a gimmick. About a half of the material we buy comes in infected with one thing or another. We had a batch of live sand recently, had both urinema and coral pathogen, Aqua roweri. It's like double whammy, both of them in one cup of live rock. Needless to say, we didn't sell that stuff. But my point here is to say that if you buy it from another supplier, it probably will be very effective in seeding a diverse community in your tank. I have no reason to think that the other suppliers have any problems with the material, but based on my testing from multiple suppliers, I expect that a non-trivial fraction of the stuff out there in the market is infected with urinema mainly. Urinema is super common in the supply chain. Yeah, so we're offering those products as ways for clients to boost their diversity and nitrifying communities. That's really the situations where I end up recommending it, is if you have low diversity or low nitrifiers. I favor the live sand if it's an established tank. I think that's a more effective way of altering an established tank. And I favor the live reef rubble for a new tank.
Yeah, because if you get your results back and your results are less than desirable, like they're not, you're missing a group or something, or low in one way or another, you can add this tested live sand to your tank and kind of boost that back up.
That's right. Yeah. And so, the report that you scroll through here, you see that these reports have in them some diagnostic information. It'll say things like, we found cyanobacteria in your tank. Here's a link to some thoughts on how to deal with that. So we try to give you some of that advice in the report and in the links. But also, for your listeners, anyone who hasn't had one of these tests done yet, you get that report back, but I also review your report myself. I review your data myself and send you a personalized email with just, my interpretation of it. And people in the reefing hobby are very, very well informed, scientifically minded people often. So it's not that I'm trying to teach you, it's that I may have spotted something you didn't. I may have spotted an oddball parasite in there. And I also have a different perspective because I look at so many of these tests. I have a perspective where I can tell you, okay, that thing we found in your tank, it's actually really common, I wouldn't worry. Or, wow, that thing we found in your tank is terribly unusual, and we should think about it. So you won't be left alone to make sense of it, I will try to assist.
Yeah, that and that is really awesome. I very much appreciate that, getting the personal email with all the other information, and it's definitely beneficial. It's really
That's good.
cool. Yeah.
Yeah, it's an interesting process. You pull up these reports and have to sit there and make sense of them.
Mhm.
And if it takes some effort for me when I wrote the code for them and spend a lot of time looking at them, then I know it must take some effort for the end user. So I want to help any way I can.
Yeah, so that's awesome. Aquabiomics, it's really cool what you're doing over there, being able to make sense of what a healthy microbiome is, and telling us if we have any fish pathogens or anything that can go awry in our tanks. And it's a new way of looking at things,
Yeah.
versus just what we can see and just thinking that it's water chemistry versus maybe something that we can't see that's causing an issue.
Yeah.
So thank you so much for joining me today on
Of course. Thank you.
Coral Reef Talk. And the best way to get a hold of you or to get their kit is simply go to the website, aquabiomics.com.
Yeah. For anything related to ordering, just go to the website, place an order. And if you run into any problems, of course, contact us through email. The website is the address is on the website, but it's easy. It's contact@aquabiomics.com. So place an order, shoot us an email if you run into any issues, and looking forward to seeing what's in all of your tanks.
Awesome. Very cool. Dr. Eli, thank you so much.
Yeah, thank you.
Thank you so much for checking out this video. If you'd like to see more videos, click this video here or this one over here, and I will see you next time on the Coral Reef Talk.